3 PowerPoints just hitting a few topics for the comprehensive final exam. So just want to stress this is nowhere near comprehensive. So you'd want to make sure to use your lecture study guides to prepare for the lecture final exam. So this was a PowerPoint we posted. This covers information from Unit 4 Metabolism, Unit 5 Microbial Growth, and Unit 6 Microbial Genetics Part 1. So those were topics that we hit on the our first online lecture exam too. So let's see if we can get this cooking here. And again, this will just be hopefully really brief. All right. So from Unit 4 Metabolism, you'll recall we were looking at how energy flows through biological systems, and we were also paying some attention to carbon and some of the other elements. And just really quickly, you guys remember the first cells that evolved, bacteria, evolved about 3.8 billion years ago, and you want to remember that Earth's atmosphere was anaerobic, right? And it wasn't until the evolution of porphyrin rings, which permitted the evolution of bacteria, chlorophyll, and chlorophyll, that cyanobacteria evolved chlorophyll A, which permitted them to carry out oxygenic photosynthesis. So slowly the Earth's atmosphere changed from anaerobic to aerobic, and that permitted this great explosion of diversity here on Earth. So again, just kind of looking at the big picture of energy flow. So we know that the sun, light energy, is a huge source of energy for life here on Earth. And again, folks, this is just like a little summary formula for oxygenic photosynthesis. So organisms that have chlorophyll A, so that'd be cyanobacteria or plant-like algae and green plants, can take inorganic carbon dioxide as a carbon source, and then they're going to use electrons from water. And in the presence of light and chlorophyll A, those electrons get charged. They become high energy electrons. And through a complicated series of events, the electrons then will be donated to the carbon dioxide. And as a result, we end up with organic molecules like glucose. So glucose is chock full of chemical energy. So this phenomenon of photosynthesis is really wild, you guys. So we're taking light energy and we're converting it into chemical energy. And then a byproduct, or another end product rather, of oxygenic photosynthesis is the release of molecular oxygen, right? And then, folks, where we spent most of our time was we were talking about ATP production, and we ended up talking about aerobic respiration. And in a funny sort of way, this almost looks like the opposite of oxygenic photosynthesis. So here our energy source are our organic molecules like glucose. And you'll recall, folks, the reason this is called aerobic respiration is that we have to have a terminal electron acceptor at the end of the electron transport chain, and that's going to be molecular oxygen. So aerobic means our terminal electron acceptor is oxygen. Respiration, as a microbiologist, when I see respiration, I know there's an electron transport chain involved. And you'll recall, you guys, this was many, many steps, right? But then kind of like our most important end products of aerobic respiration will be energy. Energy will be released. About a third of it will be used to make ATP, and 2/3 will be lost as heat. And then the oxygen will be reduced to water, and the glucose will be oxidized to carbon dioxide. So most of the metabolism unit, we were focused on chemoheterotrophs, and chemoheterotrophs require preformed organic molecules, both as a source of carbon and energy. And we want to remember that chemoheterotrophs include all members of kingdom Animalia, kingdom Fungi, some of the animal-like and fungal-like protists, so protozoa and slime molds, and then some members of domain bacteria. And folks, we could say that all pathogenic bacteria that invade another organism and cause harm, they're all going to be chemoheterotrophs. And this also helps us understand that the reason these bacteria are invading us is that they need a preformed source of organic molecules. So we're just kind of fast food for them, right? So we're going to act as the source of high energy electrons for those darling little chemoheterotrophic bacteria, and then some members of domain Archaea. And again, folks, most of the unit we focus on chemoheterotrophs, and a big topic was ATP production. So if metabolism is all the chemical reactions that occur in a cell, this is kind of like a big concept map, you guys. So maybe we could describe two basic types of metabolic reactions. Catabolism, when large molecules are broken down into smaller molecules, releasing energy. And then kind of the opposite is anabolism, where you take smaller subunits, smaller molecules, and covalently link them together. That's called anabolism or biosynthesis. And anabolism will require an input of energy. So the molecule which links the energy releasing catabolic reactions with the energy consuming anabolic reactions would be ATP. This is why we spend so much time talking about ATP production, because cells are constantly using up ATP, and that means they constantly have to be making ATP. So you'll recall, you guys, we said this is an important redox reaction to remember. So reduction is when, we said for reduction is when a molecule gains electrons, and oxidation is when a molecule loses electrons. So remember, this is that little summary formula for the complete oxidation of glucose by aerobic respiration. So remember, you guys, these are the reactants. So the reactant that's being oxidized is glucose, right? It's going to get oxidized to carbon dioxide, and the reactant that's being reduced is going to be molecular oxygen, and it will be reduced to water. And you'll recall the reason the cells do this is to release all the energy, all the chemical energy from the glucose. And again, they'll use about a third of it to make ATP. 2/3 will be lost as heat. You'll recall that for metabolism, cells use organic catalysts. And the organic catalysts we spent most of our time talking about are protein catalysts called enzymes. So since they're made of proteins, they demonstrate all the characteristics of proteins. They'll be sensitive to denaturation, so if they unfold, they can't function. We say that they're substrate specific, so if this is a globular enzyme, this little groove here, this is called the active site or substrate binding site, right? So the enzyme can only act upon molecules that can fit into the substrate binding site, so complementary in size, shape, and charge distribution. And basically what the enzyme is going to do, it's going to assist with electron rearrangement, right, of the substrate, and as a consequence, the substrates need not have such high activation energy, right? So the enzymes speed up chemical reactions, the number of end product per se second, by lowering the so-called activation energy that's required for the chemical reaction. And we said that a lot of enzymes end in ASE, ACE. We said since enzymes are proteins, we said that, again, they're going to be sensitive to denaturation. So if we looked at the number of products made per second on this graph on the y-axis, and then here we're looking at temperature, we're changing the temperature. So we said the number of end products per second would increase as temperature increases, because as the temperature is increasing, molecules are moving faster and faster. So we're going to have more collisions between enzymes and their substrate. So we'd expect that the enzyme activity, the end products per second, would increase. And then we said it reaches this optimal temperature, and then wow, activity goes way down. And this is because the enzyme is denaturing. And likewise with pH, enzymes usually have an optimal pH, the environment in which they evolved, where they can make the greatest number of end products per second. And then with pH, if we lower the pH, again, we'll get denaturation, so enzyme activity decreases. And likewise, if we increase the pH, the enzyme activity will decrease from denaturation. So just showing the process of denaturation. And then, folks, we made kind of a big deal. Now that we have this concept of enzymes under our belts, we made a we spent actually quite a bit of time talking about enzyme inhibitors, and we talked about competitive inhibitors and non-competitive inhibitors. We spent most of our time talking about competitive inhibitors, and this is where the inhibitor competes with the substrate for access to the active site. And the reason we spent so much time here is many of our antimicrobial drugs turn out to be enzyme inhibitors. And the probably most famous one is penicillin, the first beta-lactam antibiotic discovered by Sir Alexander Fleming. And you'll recall he was working with Staphylococcus aureus. One of the Staph aureus plates had a fungal spore that germinated and made a beautiful colony of the mold Penicillium. And what Alexander Fleming noticed was that the Staph aureus wasn't growing around the Penicillium colony. And so he made the brilliant hypothesis that penicillium was making some substance. It was diffusing into the agar, and it was inhibiting or killing the Staph aureus. And after a lot of work by many different people, the molecule was discovered to be the first antibiotic discovered on penicillin, right? And the reason we talked a lot about this is that we now know that penicillin acts as an irreversible competitive inhibitor of bacterial transpeptidase. And you guys will recall bacterial transpeptidase, it has to be active so it can cross-link those little tetrapeptide tails in the cell wall peptidoglycan. So if you knock out bacterial transpeptidase, the cross-links aren't made in the peptidoglycan weakens, and if the bacterium is growing in a hypoosmotic environment, the consequence will be that the bacterium will die from osmotic lysis, yeah? So we did talk about how the gram-negative bacterial cell wall, that lipid bilayer, that outer membrane, it won't permit penicillin to diffuse across the lipid bilayer. So the antibiotics have to cross through the outer membrane porins. And in most gram-negative bacteria, penicillin doesn't have the right size or shape to pass through the porins. So we said that penicillin is relatively narrow spectrum, meaning we're going to use it primarily against gram-positive bacteria and not necessarily against gram-negative bacteria, although there's always exceptions. And you'll recall that the humans tackled that problem by taking the penicillin and chemically modifying them to make the semi-synthetic beta-lactams like amoxicillin and ampicillin. These can cross through the outer membrane porins of gram-negative bacteria. So we'd say they have broader spectrum or extended spectrum. You can use them against both gram-negatives and gram-positives. And then, folks, you'll recall, and Fleming had warned us about this. Fleming said, if you overuse penicillin, you'll select for penicillin-resistant Staphylococcus aureus. And sure enough, within 10 years, that's exactly what was happening. A lot of the clinical isolates of Staphylococcus aureus causing wound infections, they were resistant to penicillin. And it was established that this earlier resistance was due to the bacterial enzyme called beta-lactamase. This is a bacterial enzyme that hydrolyzes penicillin, ampicillin, and amoxicillin. And once the antibiotics are hydrolyzed, they can no longer bind to the transpeptidase. They're worthless, right? And we mentioned that people tackled that problem. They made these beta-lactamase resistant beta-lactams like methicillin and oxacillin. And what they did with the methicillin, they just added a functional group that made the methicillin bulkier so it couldn't fit into the active site of the beta-lactamase. And I thought that was kind of brilliant, right? And then, unfortunately, you guys know now that we have MRSA, the methicillin-resistant Staphylococcus aureus, and the MRSA are resistant to all the beta-lactams because they have a mutant bacterial transpeptidase to which none of the beta-lactams can bind anymore. Yeah, those bacteria, they stay one step ahead of us, it seems. And again, folks, this is just showing, this is the beta-lactam ring, so all the beta-lactams have this beta-lactam ring. So the beta-lactamases hydrolyze that ring, and then the penicillin can't bind to the transpeptidase. And here's our methicillin, and you'll notice, you guys, they just added these chemical groups here. And that made it so bulky that the methicillin can't fit into the active site of the beta-lactamase. And that's why methicillin and oxacillin are called beta-lactamase resistant beta-lactams. It's such a mouthful. And then another approach humans used was to try to find an irreversible competitive inhibitor of beta-lactamase, and this was successful. They isolated clavulanic acid. It was made by a soil bacterium called Streptomycetes. Oops, and that's a typo right there. Sorry, that T shouldn't be there. And this big genus of Streptomyces, they're kind of famous for making lots and lots of antibiotics. They're an amazing group. So the clavulanic acid, it will bind in the active site of the bacterial beta-lactamase, so the beta-lactamase can't function anymore. And we said the first combo was they used amoxicillin, combined it with clavulanic acid. They called it augmentin. So if you were treating, say, an infection with a Staph aureus that made beta-lactamase, the clavulanic acid would knock out the beta-lactamase, and therefore then the amoxicillin would be effective at inhibiting bacterial transpeptidase. And this was just, I think this was like a bonus question, you guys. So there's another group of beta-lactam like drugs called carbapenems. They're called non-conventional. One one pharmacology book called them, you know, these are kind of like the, oh, the, the speedy sports cars of beta lactams. And the reason they're so powerful is they're really broad spectrum. They're really, really small, so they can pass through the gram-negative outer membrane porins of most gram-negative. So you could just, these were your go-to drugs, right? If you wanted a beta-lactam, say to treat gram-negative infection. And the other thing, you guys, that made the carbapenem so awesome is most of the bacterial beta-lactamases couldn't destroy them, right? Isn't that awesome, right? So for many of the multiply antibiotic resistant gram-negative pathogens of that big family Enterobacteriaceae, sometimes the carbapenems were the last antibiotics that you could use to kill them. But unfortunately, here over the last few years, and again, it's the same thing, we start overusing these drugs because they're so wonderful. So then through natural selection, we start selecting for genetic variants, in this case, which do make enzymes that can destroy the carbapenems, and they're called, those enzymes are called carbapenemases. And of course, you wouldn't need to memorize this, you guys, but some of them are called KPC for Klebsiella pneumonia, carbapenemase, again, typo there, sorry, you guys. And then the metallo-beta-lactamases, for example, the New Delhi metallo-beta-lactamases. Oh, I have to learn how to type. And so some of these gram-negatives are now referred to as the superbugs, or they're examples of superbugs, and they're said to be pan-resistant, which means they're resistant to all the antibiotics. So we don't have any antibiotics to treat these gram-negative infections, which is just such a bummer. All right, you guys, so that was kind of a quick overview of our discussion of enzyme activity and enzyme inhibitors and kind of application. So now you guys will back up and take a look at different strategies that cells used to make ATP. And we did it kind of, again, you guys, kind of historically. We talked about those first primitive cells. There was the electron transport chains hadn't developed yet. Earth's atmosphere was still anaerobic, so the first early primitive cells, they couldn't use aerobic respiration, right? So let's take a look at then how those first early cells made a little bit of ATP, right? Oops, wow. When I copied these slides, they got a little bit messed up here, so apologies, you guys. So first of all, folks, we first introduced glycolysis, literally sugar lysing, breaking in two. And you'll recall, you guys, we just hit the tidbits here. So you guys, you'll recall, usually if an organism is going to use a sugar, it's going to be glucose as a source of energy to drive ATP synthesis. So if you can see here, you guys, these 6 little balls represents the 6 carbons of glucose. And you'll recall, you guys, to get glycolysis started, we had to spend some ATP to destabilize the glucose, getting it ready to be split. So we ended up investing 2 ATP to kind of get this process started. So we had to spend some ATP, but we're going to make more ATP than we spent. Okay, and then in one of the steps, you'll recall the 6 carbon is broken into 2 3 carbons. So this is the actual glycolysis, right? And then through further enzyme catalyzed reactions, what we'll do is we're going to start ripping off some high energy electrons, donating the high energy electrons to NAD, one of the coenzymes, right, the electron hydrogen atom carriers. And with the energy released, we're going to end up making a total of 4 ATP. So we said in glycolysis, we make 4 ATP, we spent 2. So we're going to gain 2 ATP per glucose. So it's not a great way to make ATP, but in the old days, when there were just little single-celled organisms and there were huge oceans of organic molecules, it didn't matter, right? So, and then our what used to be our 6 carbon glucose skeleton, now we have 2 3 carbon pyruvates, or pyruvic acid is the end products here, right? But you guys, there was this huge problem, and that is, remember, the cell has a very limited amount of oxidized NAD. So as glycolysis is being performed, all of the oxidized NAD is getting reduced to the reduced NADH. And once the cell runs out of NAD, glycolysis stops right here. And you want to notice that the ATP is being made downstream from there, right? So this was a huge problem for the little cells. How can they regenerate their NAD? And you guys might remember The strategy the cells use was fermentation, right? And so it looks, yeah, it looks like I skipped a lot here. Okay, so let me see if I've got the fermentation slide coming up here, right? So this is where we are right now, fermentation. Okay, so here it is. So you guys, so the early solution to regenerating the oxidized NAD was fermentation. And the simplest type of fermentation was lactic acid fermentation. The pathways are often named after important end products. So here's lactic acid fermentation. You can see it's really simple. So the cells just take the pyruvate, the pyruvic acid, that's one of the end products of glycolysis. The cell's not going to do anything with it, right, in those early days. And then the pyruvic acid acts as an internal organic terminal electron acceptor, meaning the electrons carried by NADH are going to be dumped onto pyruvic acid, making the lactic acid. And the cell's not going to use the lactic acid, right? But what it needed to do was regenerate the NAD over here, right? And so we said lactic acid bacteria carry out lactic acid fermentation. Our cells, our human cells, remember how to do lactic acid fermentation. So if, for example, our cells become anaerobic, they can switch over to lactic acid fermentation for a short period of time to make just a little bit of ATP. Maybe we can survive, that anaerobic episode. So one of your colleagues a few semesters ago said, like when they're screening patients, maybe in the ER for possible sepsis or septic shock. Remember, big problems with sepsis or septic shock is you have decreased blood flow to tissues and organs. They'll look for an increase in lactic acid concentration in the patient's blood, indicating the cells aren't getting enough oxygen. I thought that was cool. We said lactic acid bacteria, they're part of our protective normal microbiome, right? The lactic acid they make on the surface of mucous membranes can inhibit potential opportunistic pathogens like Candida albicans. We use lactic acid fermentation, you guys, to preserve foods in the absence of refrigeration. We can ferment dairy products like to make yogurt or kefir or buttermilk, right? The low pH, the acids help inhibit spoilage microbes. We can use lactic acid fermentation with vegetables, right, to make pickles. Yummy. Okay, and then folks, the other fermentation pathway that we talked about was alcoholic fermentation. And the reason it's called alcoholic fermentation is this 2-carbon alcohol ethanol is produced. And we start out the same way. We start out with pyruvic acid, but in alcoholic fermentation, one of the carbons is going to get split off and released as CO2. This is why your bread dough rises. This is why if you ferment, say, grape juice or grains in a closed bottle, when you open it, the carbon dioxide will be released, so you have a carbonated vegetable, carbonated beverage. Okay, so when we lose a carbon there, we're going to end up with this 2-carbon intermediate acetaldehyde, and then this becomes the internal organic terminal electron acceptor. And again, NADH is going to dump its electrons, its hydrogen onto acetaldehyde, making the ethanol. This is what the cell is after, the NAD. And we said that alcoholic fermentation is another way that our ancestors, and even today, we can preserve food in the absence of refrigeration. Because remember, alcohols are denaturing agents, so they can inhibit the growth of spoilage microbes, right? By the inhibitory ethanol. So you recall, as we're going through time, and these little cells are doing this fermentation, which is very wasteful, right? The fermentation end products like the lactic acid, the alcohol, it's still chock full of high energy electrons, so it's very wasteful. They're only gaining 2 ATP per glucose, right? And the fermentation end products like the alcohols and acids, they're inhibitory, right? So as cells are fermenting along, they're using up their preformed organic molecules, and now we're getting higher concentration of cells. So there was natural selection for genetic mutants who could make more ATP per glucose, and using a process where the end products weren't so toxic, right? So these mutants evolved aerobic respiration, which is just phenomenal, right? And so we said a big key to aerobic respiration was the evolution of porphyrin rings. Porphyrin rings were essential for the evolution of bacterial chlorophyll and eventually chlorophyll A. And you guys will remember chlorophyll A permits oxygenic photosynthesis. That's how Earth's atmosphere was switched from anaerobic to aerobic. So that was important for aerobic respiration. And furthermore, The porphyrin rings were essential for the evolution of cytochromes, and cytochromes are essential parts of electron transport chains, right? So for aerobic respiration, again, as a microbiologist, when I see respiration within a cell, I think an electron transport chain is involved. So those porphyrin rings was really a big deal. When they evolved, that really set the stage for the evolution of aerobic respiration. Now, what's the big deal with aerobic respiration? What makes it so different from fermentation? Well, you guys will remember that we're going to rip off all the high energy electrons from, for example, glucose, and we're going to have our hydrogen atom electron carriers, NAD and FAD, accept those high energy electrons, and then the NAD or FAD are going to deliver those high energy electrons to the electron transport chain. As the electrons are passed from one member of the chain to the next, energy is released, and the electron transport chain uses the energy released from those high energy electrons to pump protons across a membrane, forming a concentration gradient of protons across a membrane. And we know that's a source of potential energy. That acts as a little battery almost. So we talk about the proton motive force, right? And then it's like, so what's the connection between the electron transport chain and forming the proton gradient and ATP production? Well, then you guys will recall the key is this other membrane protein complex, ATP synthase, is located in that same membrane. It has a proton channel through which the protons can now flow from a high concentration to low. And as they're flowing through, it's almost like a little electrical current, a little positively charged electrical current. It causes shape changes in ATP synthase, and this helps drive the electron rearrangement that permits ADP to be phosphorylated to make ATP, right? And we said a massive amount of ATP is made in this process called oxidative phosphorylation, like about 34 of the 36 to 38 ATP are made using oxidative phosphorylation. So this was huge when the electron transport chain, the proton gradient, and ATP synthase evolved. This is what permitted massive amounts of ATP to be made from the high energy electrons of glucose. And again, folks, just a reminder that the evolution of the porphyrin rings were really important. And this is our, and again, you guys, sorry, these slides got a little bit messed up when I copied them. So this would be our summary reaction, you guys, and this arrow should be right there for aerobic respiration, the complete oxidation of glucose using molecular oxygen as a terminal electron acceptor, right? So remember, all the high energy electrons will be stripped off of glucose, and they'll eventually get past the electron transport chain to help drive formation of the proton gradient. So the glucose will be oxidized to carbon dioxide. O2 is our terminal electron acceptor. At the end of the ETC, it's going to accept those tired out low energy electrons, so the O2 is going to get reduced to water. And with the energy released, the energy, some of the energy, about a third of it's harnessed to make, this is a hypothetical max, you guys, they don't think this actually happens, but we're going to use it anyway to keep our life simple. So this hypothetical max is about 36 ATP per glucose in eukaryotes and 38 ATP per glucose in prokaryotes, and then the other 2/3 is released as heat. So complete oxidation of glucose requires electron transport chain to create the proton gradient. Requires an aerobic environment. O2 is the terminal electron acceptor. And folks, you'll recall that there were two ways to make ATP. So substrate level phosphorylation, this was how low levels of ATP were made, a gain of 2 in glycolysis and a gain of 2 in the Krebs cycle per glucose via substrate level phosphorylation. And then the massive amount of ATP made using the ETC, proton gradient, and ATP synthase. We refer to that as oxidative phosphorylation. The use of a chemical concentration gradient across a membrane to drive cellular work is an example of chemiosmosis. And furthermore, you guys, more advantages, the end products of aerobic respiration are far less toxic than the alcohol and acids of fermentation, so we're going to get CO2 and water. And another thing that we just really briefly mentioned were the intermediates in aerobic respiration can be siphoned off and used in anabolism. So for example, amino acid synthesis or nucleotide synthesis. Yeah. So on the final exam, you guys, it'll probably be something kind of like matching, like match the end products of glycolysis, match the end products of the Krebs prep, match the end products of the Krebs cycle, right? So, and I know that memorization is just brutal, but it'll probably be, yeah, that's probably how you'll see it on the final exam. And again, this is just showing you the stages in aerobic respiration, how many ATP are made, just a big overview. And then, folks, we just really briefly talked about a more primitive type of respiration that evolved before aerobic respiration. Remember when Earth's atmosphere was still anaerobic? So in anaerobic environments, microbes, they evolved the ETC, but there's no oxygen available, so they evolved anaerobic respiration, so no O2. The electron transport chain involved in anaerobic respiration is different than the one for aerobic respiration. So in bacteria like E. coli that can do both anaerobic and aerobic respiration, they'd have two different electron transport chains. In anaerobic respiration, the ATP yield is less than aerobic respiration, but it's still a lot higher than fermentation. So in anaerobic respiration, a molecule other than oxygen is a terminal electron acceptor, and we talked about how nitrates could be used. And this was part of the nitrogen cycle. You guys remember how we said under anaerobic conditions, like if your fields are flooded with water, the soil bacteria, now it's anaerobic environment, they can switch to anaerobic respiration using nitrates as a terminal electron acceptor, reducing the nitrates all the way back to molecular nitrogen. And plants, nor can we use molecular nitrogen, is a source of nitrogen for amino acids and nucleic acids. So this is called denitrification. So if you're a farmer, you wouldn't want it if you're trying to conserve nitrates in your soil so your plants can grow. But if you were, say, working in a sewage plant, and sewage plants are fascinating, guys. There's so much cool microbiology going there. Before you release the sewage into the rivers or coastal waters, you want to get rid of usable nitrates, right? So in a sewage plant, you'd actually want denitrification to occur, right? Because if we release the sewage into our waterways or coastal waters and they're rich in usable forms of nitrogen, like nitrates, that will cause those harmful algal blooms, those HABs. And we can have, even though they call them algal blooms, it can include cyanobacteria. And the reason those harmful algal blooms are bad is there's some toxin-producing strains of cyanobacteria in algae. One of the classic ones is paralytic shellfish poisoning caused by a specific type of algae. And furthermore, if we have an algal bloom, not only can they make toxins, but when they die, then aerobic respiring microbes are going to tear their bodies apart, use them as a source of carbon and energy, and they'll use up all the oxygen in the water. And so then you have an anaerobic aquatic habitat, and most things don't grow there very well unless maybe you're a anaerobic bacterium. So they talk about dead zones being produced, right? So that's why as a sewage treatment operator, you actually want denitrification to occur. We talked about how sulfates can be used in anaerobic respiration. Sulfates will be reduced all the way to hydrogen sulfide. You might recall the TSI, the triple sugar iron slants that we introduced in the lab modules. Hydrogen sulfide, it smells like rotten eggs, and we're normally kind of repulsed by it. And it's also toxic and bind to our cytochromes and act as cytochrome inhibitors, right? So, so if you visit kind of a marshy area and it smells like rotten eggs, a lot of times that's this hydrogen sulfide being produced. And we didn't go into CO2 or carbonates that can be used as terminal electronics that we probably should have, because this is really bad, you guys. The product of this anaerobic respiration is methane, and methane is a really important greenhouse gas contributing to global climate change, right? So, and then, folks, we did mention that some bacteria, like our good friend E. coli and many of its cousins, the Enterobacteriaceae, they're so versatile. If there's no O2 present, they can carry out fermentation or anaerobic respiration. If there's oxygen present, they'll switch over to aerobic respiration. So that versatility, remember you guys, adaptability, that's how organisms survive, is being adaptable. So we can see why E. coli is so successful. And then just really quickly, you guys, we said the earliest form of chlorophyll to evolve was called bacterial chlorophyll. And this permits anoxygenic photosynthesis in that no O2 is made. And bacterial chlorophyll will use carbon dioxide still as our source of carbon to make organic molecules, but instead of water as an electron donor, we're going to use hydrogen sulfide. So again, in the presence of light and bacterial chlorophyll, electrons from hydrogen sulfide are energized in a complicated series of steps. They're donated to carbon dioxide, so we end up converting our light energy into the chemical energy. And then as we have oxidized hydrogen sulfide, we're going to end up with elemental sulfur, right? And here's these cool purple sulfur bacterium, and these little yellow refractile granules are actually collections of sulfur inside the cells because they've been carrying out anoxygenic photosynthesis. And then we said with the further evolution of chlorophyll A, this is what permitted oxygenic photosynthesis, in which water acts as electron donor. In the presence of light and chlorophyll A, electrons are energized, donated CO2, making organic molecules like glucose, rich in chemical energy. So again, we've converted light energy to chemical energy, which is so amazing. And the reason it's called oxygenic is when we oxidize water, we end up with a molecular oxygen. And so this is the process that permitted Earth's atmosphere to be converted from anaerobic to aerobic, which is pretty amazing stuff. The nitrogen cycle, again, you guys, just really quickly, we didn't go through all of these steps. We'll just go through 3. So remember, you guys, most of the air we breathe is rich in molecular nitrogen, 2 nitrogen atoms, triple covalent bond. The those triple covalent bonds so hard to break, right? So very few organisms make the enzyme complex called nitrogenase that can convert the molecular nitrogen into a usable form of nitrogen, ammonia, right? So this process is called nitrogen fixation. It requires the enzyme nitrogenase. And again, very few organisms make it, but cyanobacteria make nitrogenase. And then we also talked about, you guys, the rhizobium, the soil bacteria that have this cool symbiotic relationship with plants known as legumes, so peas, beans, clover, peanuts. The rhizobium invade the roots. A plant makes a little anaerobic root nodule for them. And in the root nodules in the rhizobium use nitrogenase to fix nitrogen into the ammonia, which the plant can use. And then it's so cool, you guys, because nitrogen fixation takes a lot of energy. So up here, the cool little plants, through photosynthesis, they're making sugars, and then they transport the sugars down to the bacteria, where the bacteria can use the sugars to make ATP. So this is win-win, right? I love win-win situations. So this is an example of mutualism. And then, folks, we said, furthermore, in the soil, there'll be other kinds of bacteria that will take the ammonia and can convert it into nitrites or nitrates. This process is called nitrification. My understanding is plants really like nitrates, right? So if we were farmers, you guys, we would love nitrogen fixation. We would love nitrification, right? Because this is all forming usable forms of nitrogen for our plants, for proteins and nucleic acids. But we said that, as we mentioned, guys, if a farmer maybe over irrigates their fields, or maybe there's, weeks and weeks of rainstorms, waterlogged soil is anaerobic, right? Because oxygen doesn't diffuse well through it. So now you have an anaerobic situation. So your bacteria can switch to anaerobic respiration, take the nitrates, and in the process called denitrification, convert them all the way back into molecular nitrogen. So again, if we're a farmer, maybe we don't want to do that. If we are working in the sewage treatment plant, we do want this to occur right in the sewage before we release it. And also, farmers might want denitrification if they have excess water running off their fields that might have excess nitrates in them, right? We don't want excess nitrates getting into our waterways because it will cause those harmful algal blooms. So you'll recall that some farmers are making bioreactors where they run the irrigation water through. Sometimes they're just holes in the ground that they've dug, and maybe have like wood chips in there, so the denitrifying bacteria can form biofilms, and so then you run your runoff water through the bioreactor before it's released into the rivers or the coastal waters, and that too will help reduce nitrates and thus reduce harmful algal blooms. So that's a really cool way, kind of working with nature. And then, folks, I think what I'll do is stop this audio here. because we still have microbial growth, which isn't that long, but then we have microbial genetics. And I know, they tell us as teachers, don't make your videos too long, and I always make these really, really long videos. So let's stop here, and I'll just label this as the metabolism part of the review here. And then, folks, will do another audio for microbial growth and microbial genetics, part 1. Yeah, okay.